So after loading with some of the packages, I am proceeding with, suppose yesterday we did the practice session for TCG head and neck cancer, and now I am proceeding with some other type of cancer. So this is STAD, which stands for stomach adenocarcinoma. So once I prepared the GDC query and prepared the file for downloading in the same manner which I did for HNSC, I will be preparing the missing samples again and removing the missing samples and downloading the files in the matrix format like we did for the HNSC. The procedure is same. So as you can see, there are a total of 192 columns or 192 samples are there. And this is segregated fully of around 60,660 entries, which are the ensemble IDs. Now this you can see like 11A is the normal IDs for the sample. Now I need the metadata information, which I will be downloading in case when the data is fully normalized and clean. I will be needing this for probe to gene mapping, because in here actually no sequencing platform or other things are there. We need the metadata information, and for that we first need the raw data from the data clean, and we are storing this in gene metadata. So this I'm getting the information like there are various information like gene ID, gene name, level, and we need this gene name column, and we need gene ID, gene type, and gene name. So gene ID is the same, right? This is the gene IDs. I need the ensemble IDs. Gene type I need because I need only the protein coding. And as you can see, there are many types of gene types, like apart from the protein coding, like LNC RNA and transcribed unitary pseudogene and unitary pseudogene are there. So we need only the genes which are actually protein encoding and also the corresponding gene names. So I need these three columns. So I will be fetching this. And for the batch correction, I need the tissue source site and the plate information. So first I will be fetching all the call data information from the call data clean. And this I can fetch various information from here like TSS, plate, and as well as the, after fetching the DFH, which is the get IDs from the data clean. So I will be fetching the tissue source site and plate information from this. Now I will be pre-processing and log transforming basically my count values, and I'm feeding in the matrix file first and the column data. So column data is just the information where I have the information regarding the samples regarding which, whether it's protein or whether it's tumor or normal. So I'm defining the tissue type as the design matrix, and as you can see in the design matrix, it's stored in the column data file. There are various information available in here. So this is the sample type like primary tumor and solid tissue normal. And this is the tissue type. I need the information regarding whether it's simply a tumor or the sample, or is it normal. I can see 1, 2, 3, 4, 5, 6, 7, 8, 9 normal samples are there out of total 192. So I will be pre-processing this whole information, and the counts let which are having an average count of less than 10, I will be removing, as these are the low count IDs. And after that, I will be performing the batch correction using the batch factor as plate and adjusted adjustment as the TSS, which is the tissue source site. Once that has been done, I will be transforming my matrix in the same manner as we did for the microanalysis for converting all my expression values in the batch corrected file in the Z-score expression format. So once that has been done, I will be using the index matching and inner join function. And if you remember, we have the already gene metadata information with us. in the Z score, and we will be using that to map the probe or run the sample IDs to their corresponding gene symbols. And I need only the protein coding ones and not the others. I will be deleting the others. Suppose that I will be doing the aggregation for removing the duplicate or the multiple or the repeating genomic entries regarding the gene. So once that has been done in the AGG file, you can see there are two additional columns apart from the 192 columns which must be present. So this is NA and NA because once they have been mapped in the first column, the last two columns have been made empty. So we need to eliminate or remove these additional extra columns. After that, I will be checking for the officially approved gene symbols via the NGNC package or the TCG biolinker package in the same manner as we did for microarray. And after that, removing the finalized, not available or missing entries in the AGG filtered file. So we started from 60,660 genes. And after removing the lowest 25% variance Gs in fill 2, now we are actually left with only 14,000 genes with us, which are unique and fully pre-processed. So we can plot the final QC plot at the end of the lowest variance 25% gene removal, and we can plot these QC plots at many steps which are in between, like we did for the microarray as well. But for the convenience, we are actually plotting only at the last step. So as you can see, the density plot is also in the ideal condition, which should be. Now we'll be preparing the design matrix as per the tumor and normal information and using the LM-fit model of lemma matrix to compute the p-value and adjusted p-values. So after that, I will be saving this file in my folder name as dex.txt. So this is the final file. I will be adjusting the column headers perfectly that should be present in the actual condition and renaming the first thing as genes. So after getting bold all the headers, I will be taking the condition of adjusted p-value less than 0.05. I will be saving this file, and I don't need this average and t values, so I will be deleting them. And I will be creating an additional column as same like ABSFC, and use the formula of absolute of the fold change. After entering, I will be computing the absolute fold change value of all the log fold change values. Now arranging this in the decreasing to increasing order, and conditional formatting highlight the sense greater than 2. So for the 2, you can see the values or the genes are ending up at roughly 1100 genes. If we say to be accurate, it is around 1132. I copy this, and I need these 3 columns only: log FC, p value, and adjusted p value. Clear the reads from the entire sheet, and I will be saving this file in the txt format. So this is the DEGs at be it adjusted p value 0.05 and log FC2. So we have to load this file again in the Excel or in the R command, and the final is degs 16 sept. We're loading this file, and after that I will be just fetching the expression values of these particular tags which are 1132 from the filt2 file. And actually, basically, now we are doing the survival analysis. So for the survival analysis, we don't need the normal information and only need the tumor information. So in the filtered column, I will be taking only the tissue type, which is with respect to only the tumor samples. And in the filtered, as you can see, it has fetched me the logical indices or matrices. So in the SURT, SURT, I am taking the expression values of only 184 out of 192. So it has removed around 8 normal samples for us. Now we shall be opening HGNC homepage, which is the official homepage for checking any kind of protein or gene families. So suppose we are interested in, for the task today, for the Chromobox family. This is a class of protein families, and the official gene members or the protein members can be taken from this officially approved NIH and NCBI approved HGNC homepage. You have to click on the approved and the protein coding genes. So these are the members of the Chromobox family. So CBX 1, 2, 3, 4, 5, 6, 7, 8. There are a total of 8 members. So whatever protein or gene family you need to take for the consideration, you can take from a GNC homepage only. So since there are 8 members, I will write that, and I will make a vector first in the quotes and the comma, like CBX 1, 2, 3, 4, 5, 6, 7, 8. And I'm asking like actually which indices or which row names actually all these 8 members are present in my data set. So it has returned me out of these all 8 members, only 2 of the gene members are present. One is at 149th position and one is at 150th. So we shall be checking the survival analysis of both these genes one by one. So first is suppose at 149. So we need to uh, clear the index for that, and we'll check for the 149. It's actually CBX7. So I'll be preparing the expression files first, and basically I need 3 4 types of expression values for that. So first I need the sample IDs and the expression values of only CBX7 from the main file. And one thing I need, because after preparing this file, I need some of the survival information for further doing the analysis. So I am checking whether in the columns, is there any columns related to vital stats, days to last follow-up, and days to death. So it's returning that it's presented 31st, 53rd, and 59th position. So I need only these three positions for us. So suppose first is the vital stats, right? So vital stats, we are getting in the information like that, like some patients are alive and some are dead. So I am using a if else statement and I am storing additional column name as diseased. And I'm saying that if the vital statistics is alive, like the event is not happened, so that means just put a binary operator as zero. If the event has happened, then the binary operator must be one. Event is the death, so the death is the event and which should be represented by one. So after that it has been done, I will be just coinciding the alive and the dead as 0 and 1. So alive patients are 112 and dead are 71 out of total. Once that has been done, I will be taking the overall survival time information from the days to last follow up till the patients were coming for the last follow up. That can be even after chemotherapy or once the total time has been counted from their treatment. And I will be storing this information and C binding this information again in the F3 vector. So this is the OS. Disease is the alive and the dead time. And whosoever are basically alive, I am taking the count till they came till the last follow up. And one is for the death. So 422s, 422 days are is counted till the patient has been died. And suppose for this patient, 229 is the time. For this 185 is the time. And suppose zero is the alive. So we don't know the particular time of the patient as he or she didn't came of the for the last follow-up. So they have been dropped from the clinical trials. So I will be renaming these columns as time and status. And again, if checking, so time is the information status and third is the expression value. These three values we need for the survival analysis. So we can save this file also. So I will be preparing two additional values like high expression and low expression as plus 1.96 and minus 1.96 based on the hypothesis, null hypothesis testing of the 0.05 at both sides of the tail, if you consider, because our values are actually in the z-score. If you remember, we did the pre-processing for the z-score transformation. As per the null hypothesis, the values correspond to 1.96 on both sides of the magnitudes. So suppose is for the first is for the CBX7 as we are doing. So we are preparing a if else statement. So in the F3, we are saying if the expression value of the whatever the CBX7 is greater than or equal to the higher expression value, which is 1.96, assign that is high. Or otherwise, if the value is less than or equal to low expression, so for the less than, assign low, and if it's equal to that particular 1.96 value, assign as mid. We run this, and after that, we assign three levels like mid, low, level, high, high. And after that, we plot the survival plot for the using the gdsurf package. And as you can see, first we need to check in the particular expression pattern. And as per, firstly, we need to check the p-value. So the p-value must be 0.005 less than 0.005. So it's perfectly less than 0.005. So the survival trend between the low, mid, and high expression status of the patients is in perfect statistical alignment. Now we want to check whether it's actually perfectly matching with the survival status of the patients or not. So first we need to check the log FC value of CBX7. So CBX7 is a tumor suppressor gene as it has a negative fold change. So it's not an oncogene, it's underexpressed or a tumor suppressor gene. So the plot must also say like that. but the plot is having opposite trend. As you can see, the blue line, which is for the high. So it's saying that if the this particular gene is having the high expression, the patients in the high expression group will die or decease first, as the curve is dropping st- instantly before the low one. Low one is represented by the green line, and after that, the mid is after that. So the trend should be perfectly matching. If the green line dropped first, before that, the blue one, I hope you understand, because it's a tumor suppressor or underexpressed gene. So high expression gene group or pattern must not drop down first. It must be the green one, which represents for the low pattern. So first we need to check the p values, obviously, and after that we need to check whether it's perfectly in line with the survival trend or not. Suppose that since there were two members, so we'll be checking for the other ones also of the CBX family members. So one was at 149 and one was at 150. So for 150, we'll be again checking, and the CBX member is CBX 8. We need to replace the CBX 7 with CBX 8 here. And if you plot the plot for CBX8, it's statistically insignificant, as we see the p value is greater than 0.05. So this is the first line of rejection or acceptance, where we reject the graph based on the statistical p value less than or greater than 0.05. So this is how the survival analysis is generally done using the R scripts. And this is the overall survival analysis. Now we'll show you how to do the survival analysis using the databases in case we don't have the survival data with us. You can type KM plotter, and after that, you can see the mRNA gene chip is available for these types of cancers. So this is for the microarray, and for the RNA-seq, you can see these types of cancer are available. For all the types of cancer is mostly available. You can click on pan cancer. After that, you have to go in the plot options, generate high resolution TIFF file, invert the hazard ratio values before one, and compute the median over an entire data set. Now you have to deselect all the files. And suppose it was for stomach adenocarcinoma, which we were doing. And you have to just auto select the best cutoff option. Apart from that, the values can be bifurcated or segregated based on the median also, which is generally used in case of survival analysis. But we did not on the median or the 50% range, since the values were z-score transformed. The KM plotter has its own values. So suppose you want to check for any of the gene of your interest, like suppose it's ANLN, and you want to check the overall survival analysis. So we can check the plot. So as you can see, it's nearly a significance level, but it's insignificant anyways. So as you can see from the plot, the black line is having a drop at the 50th percent probability. It's declining first as compared to the red line. So black line is for the low expression. You can also match from the table. So if suppose this expression is having low value, so there will be the death of the patients earlier, because these patients will be alive for 26 months if that particular gene is low. But if that particular gene is high, so in the high expression groups, the patient are living for 56 months, which is nearly around double of this, more than double of this. So you can either check the median survival table or the graph In case you are having any confusion of the graph, the table helps to check for the alive status in the months, right? So it's insignificant. Once the value is significant or below 0.05, you have to just check the table. And if you have the expression status with you in the dex table, and it's perfectly matches with the trend, like if it's low expression and the patients are dying in this, the gene must be also underexpressed or down regulated. And if it's the opposite case, that means the gene, if oncogenic or under higher expressed or overexpressed, then the trend must be opposite, like the red line must be declining first as compared to the black line. So after that, we shall be doing the C bioportal or the mutation analysis. You have to click on C bioportal on the Google, and you can have a look of the multiple cancers here. So we shall be doing for the STAD, which is stomach cancer. You can check for the stomach here. If you cannot find, you can type here also. So as you can see, the stomach is here. You have to click on the TCGA Firehose Legacy. It's the official TCGA sample repository from here. So stomach cancer, and you have to query by gene and explore selector studies. So you have two options. If you have the gene list with you, we have the gene list with us, but we will do it for the samples for which we did the differential expression analysis, if you remember. So what I will do, I will save this file named as F3, and just saying that F3 is having the file because I need the sample information for us. So it's saying like tumor only, and I will save this file first. So after that, I will be opening this file in Excel because I need the same number of or same exactly same number of 180 to 184 tumor entries for which we did the differential expression analysis. So the tumor entries are actually here. We need to copy this and paste it in new file. And now remove this while number at the end, like 01A and 11A. 11 is not there. There will be only 01A because we removed already the normal information, if you remember. So I will be removing this while number from here. I can use text to column and use the fixed width option to just remove the 01A. After that, once it has been removed, I will go to C Bio portal, and in the explore selected studies, I will click the option, and you can see there is custom selection here, right? You have to click this and copy this all sample IDs from here and paste it here and filter to the listed sample. So out of 478, which was initially present, now we are the same 184 for which we did the differential expression analysis. So I will be just writing the genes of CBX members one by one in the row, in the basically one by one in the line numbers CBX1. So once that has been done, I will be clicking on query. And as you can see, I will tell you, we can get information like all these 8 members are altered in 30 of the patients. So 30 patients of 184 comprise 16% of the samples. So less than one fourth of the total 184 is being altered total. And you can see the highest alteration or the frequency or the mutation is reported for CBX4, which is 6%. So what this 6% mutation is actually, you can go to cancer type summary, and after that you can go on cancer study, right? And after that you can click on CBX1. So after placing the cursor on the bar plot, you will see one case like that is altered for deep deletion, showing deep deletion, which is which is the color for the blue. Amplification is for the red, as you can see at the bottom, and green is actually the missense mutation, where one case or one patient is being altered. Amplification, you can see the four patients are altered for respect to CBX1. So if we see CBX4, which was having the highest mutation, you can see five cases for amplification, cases for missense mutation, which is in green, and in the blue you can see deep deletion, one case is being altered. So you can check for each one of the family members of the proteins one by one, and you have to next click on the mutations, and here you can see the pfam protein domain mutations. Here this is the basically the amino acid bar, and you can see the R30th position, or which type of position is actually there for which of the protein members. As you can see for the CBX4, if you see, so this is the missense mutation. So you can see A540V. So it's showing that at the 504 position, instead of A, which is alanine, valine has been replaced. And at the 230th 6 position, in case of alanine, T has been replaced. So T stands for another amino acid. Likewise, P, which was originally present at the 472th position, L has been replaced, which can be leucine. So you can check which kind of mutations are there. Like these are the missense, and suppose this is the in-frame deletions, right? So for which type of this is the frame shift deletion, like the frame have been fully shift. For the K106 RFS 31st position have been replaced for that. Likewise, for all the types of different uh, gene members or the protein members, we can see the type of the mutations here, and the overall summary has been denoted by the table at the right side. You can also check one of the important external validation plot in the bioinformatics called GPR2. So it assists in checking in the expression status as well as the stage plot analysis in case we don't have the expression values with us in our hand. Apart from this, first we will check the timer database. You have to click on the timer database to check for the tumor infiltration mutation analysis. So timer 2.0 is the database. You have to click on the immune association. And in the gene, you have to type, suppose CBX4 was having the highest mutation of all. So you can check the correlation with this with any of the members like CD cell, CD8 plus T cell, CD4 plus B cell, neutrophil, monocyte, macrophage, dendritic, T cell, natural killer cells. So, and obviously the macrophages are also there, which can be bifurcated in M0, M1, and M2 based on their levels at like it is assisting or actually helping to destroy the tumor cells. So once we submit, you can see suppose for the STAD, which is here, or you can check for any of the cancer here. Like if you see for PEAD, so PAAD is actually the PRAD is the prostate cancer, and this is pancreatic cancer. So for the T cell timer for the pancreatic cancer, you are seeing a negative correlation or a negative trend. Like if the infiltrating levels or the mRNA levels or CBX4 increases, the T cell CD8 plus will decrease, and the relation is actually significant as the p value is 1.4 into 10 to the power minus 2, which stands for 0.01, and the R, or the Pearson correlation value, is minus 0.13. Also, the tumor purity information is showing on the left side. Apart from that, you can check for the other types of immune cells. Like suppose we can check here for the macrophages. So you can macrophages, you can take the absolute value of M0, M1 is written like M1 cybersort ABS, M2 cybersort ABS, or you can in general take the macrophage timer expression also. Like for the lung adenocarcinoma, you're seeing a negative correlation with respect to the macrophages, and in case of LUSC, it's positive. So both the different types of lung cancer are having a inversely related proportional with respect to the CBX4 gene. So likewise, there is a database called GPR2 where you can check the box plots. This is the same box plot or the expression plots. If in case we don't have the expression value with us in our hand, you can take the log fold change cutoff as 1 or 2 or anything, and the p value must be fixed at 0.05. You can take for all the cancers or any of the specific cancer for which you are saying. Like for LUAD, if you check, you can click on add and then click on plot. After that, the plot will appear and you can just download and open that in the PDF file. And it's not statistically significant, because if it's statistically significant, you will be seeing a asterisk mark at the top showing the p-value of less than 0.05. Apart from that, you can check the stage plots. Survival analysis is also possible here, but it's preferable to do from the KM plotter only. And the stage plots check gives you brief information of how the tumor stages are actually behaving with respect to the mRNA expression levels of the gene. So this is the session for today, which we did for RNA-seq analysis and saw some of the external databases for the bioinformatics validation. And this can be helpful for the research purpose and the research paper publication. So if you have any doubt regarding the session, please do let me know. And how was the session useful to us? And you will be using these tools and other things for your research purposes in the future.